Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. Joseph Buxbaum will present Autism and New Findings. The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, the Foundation has awarded more than $342 million. 100% of all donor contributions for research are invested in grants to scientists who are working to find breakthroughs in disorders such as ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, OCD, post-traumatic stress, and schizophrenia. I'm delighted to introduce Dr. Joseph Buxbaum. Dr. Buxbaum is a professor in the departments of psychiatry, neuroscience, genetics, and genomic sciences, vice chair for research in the Department of Psychiatry, director of the SIVA Autism Center for Research and Treatment, and chief for the Center of Excellence in Neurodevelopmental Disorders at the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai. Dr. Buxbaum is a member of the Scientific Council of our Foundation, which identifies the most promising research ideas to fund with our NARSAD grants. Today's webinar will begin with Dr. Buxbaum's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions throughout the presentation. Following the presentation, I'll present your questions to Dr. Buxbaum and will address as many of them as possible. And now, I'm pleased to present Dr. Joseph Buxbaum. Joe, the stage is yours.
Thank you, Jeff. I need to take 1 second to figure this out. All right.
So Dr. Buxbaum, if you can accept to share the screen.
Yeah, I've done that.
For now, just 1 second, if we can move. That's it. Excellent. Thank you.
Okay. All right. Well, thank you, Jeff, for the introduction and for the invitation. I'm very pleased to be able to present some of the work that's going on in autism today that I consider really groundbreaking. And it's a fairly dense topic, and it's hard to encapsulate. So I know the listeners are going to have you'll have to hold on to your seats. So I do want to start with a disclosure, which is I will be talking a little bit about a potential treatment for autism, and there is a patent that Mount Sinai and I share on this. So there's no money changing hands, but just so you know. So, and I want to start with a little bit, I want to start with my thanks, because I often rush them at the end. We have a lot of support from in our autism research from multiple foundations, participating families, of course, and known and anonymous donors. And the folks in this picture are people from the SIVA Autism Center, which is uses a model which is becoming more common, which is that you have your basic scientists, your clinician scientists, and your clinicians all working together and trying to kind of move things along as quickly as possible. So what I want to kind of begin with is this concept is that there are multiple ways of looking at any disorder, any developmental disorder in this case, and I'm going to call them top-down versus bottom-up. So top-down is kind of the manifestation of the disorder, and in the case of a behavioral disorder, it is the behaviors that are being considered. So I will not really go into too much explanation about what is autism, but I'll just remind people that autism is defined by having deficits in social interaction and the social use of language, and the presence of stereotypical mannerisms and or prescribed interests. So those are behavioral things. They're not, they're not a cause of anything. They are what you can observe. And so the bottom up is where you try and understand what are the causes of a particular disorder. And there's value to looking at both sides of autism. Certainly, the first-line treatment for autism is behavioral, and some of the very effective things like the Early Start Denver model or ABA therapy. That's based on monitoring behavior and watching for change. Unfortunately, the bottom-up approach, which is thinking about the causes of autism and what can be, how that can relate to better interventions, still has not yielded fruit, but we're getting very close. And so in autism, a major cause is genes. And in fact, what we know most about autism in terms of causes are genetic causes. And so my talk is going to be based on this idea, and I'm calling it a hypothesis because it formally still is, that as we learn more about the causes of autism, we'll do better and better with treatments. Okay, and I'm going to use this slide a couple of times, and it'll help frame why we spend so much time finding etiology or causes of disorders. And in the bioethics space, people talk about benefits to patients, families, and society. And we do believe that as we discover causes of autism, it will lead to more informed care and personalized medicine because it's going to give us new targets for medicines and ultimately new medicines. Benefit to families, I'll talk more about with specific examples, but a family that gets a genetic diagnosis, for example, in autism, can know more about how it's going to develop over time, knows about recurrence risk, what's the chance of having another child in that family with a similar disorder. And this is something that I consider very important, which is not actually part of bioethics literature. When a family finds out something about a cause of a disorder in their family, they become very active about trying to promote research and raise resources to study that aspect of the disorder. I'll come back to that later. But mostly I'm going to talk about this benefit that's a little bit more abstract, but I think very important, the benefit to society and ultimately back to the patient. Okay, so I'm talking about genes and genetic effects, and by way of very brief background, we have about 22,000 genes in our DNA. And each gene codes for a different protein, and these proteins are the units of function in the body. So one protein many of us know about is hemoglobin, and hemoglobin is a protein that's able to carry oxygen where it's needed. And we have many, many hundreds of thousands of different proteins in our bodies, each with a different function, and they're coded for by about 22,000 genes. And those genes can have various mutations or variants in them, and that's what makes us all different and unique. And so when we talk about genetic variation, that different flavors of each gene we might have, it turns out to be very useful to think about the frequency of that genetic variant and its effect size. What that means is some things that humans have are quite common. There are some things that specify eye color or hair color or even certain diseases where the gene variant that underlies that many of us have. And then there are some flavors of genes where the gene variant is so rare that only a few people, or even just one person on the planet, may have that particular variant. We call that a private mutation. And each of those genetic changes can have a tiny effect. Like, for example, there are many, many genes that work together that determine your height. Each one has a very small effect by itself. Or it could have a very high effect size. It could increase risk, let's say, by 50-fold. Okay? So when you plot, you kind of make this schematic diagram of the frequency of the genetic variation or the allele and how much of an effect it has, we can tell you with a lot of certainty that we're not going to find autism genes over here. And the reason is because if something is both common and has major effect, in other words, really increases risk for autism, it's not going to be maintained in the population. It's going to be purified away. That's a fancy way of saying that Darwin was right. Okay, and in fact, most disorders that happen, early in life, that are, profound or significant, do not have genetic variation that's common. And that's simply because it's selected against. The example that we often use for a common genetic variant that has high effect sizes is ApoE in Alzheimer's disease. So there is a genetic variant, a gene flavor that is found in about 20 to 30% of the population of the world that increases risk for Alzheimer's disease between 3 and 10-fold. So that's a high effect gene. And it exists in the population. 20 to 30% of the people have it. But the only reason that that can exist in the population is that Alzheimer's disease occurs after the age of reproduction. So it's not subject to Darwinian purification. So we will not find autism genes that are both common and have major effect. Genes that have mutations that are very rare and have tiny effect, we're not going to find them only because we don't have enough samples. If something has both a tiny effect and is very rare, it's going to be almost impossible to find. So we're pretty clear on autism and related disorders that what we're going to find is in this middle path of rare things that have high risk and common things that have low risk and maybe something in between. And practically speaking, it's very useful to divide this and look at these two parts, these two arms separately. So let me just go back. I'm going to start with this part of the curve over here. Common things that maybe everybody has a few of, but have tiny effects, increase risk by 10% or less. Okay. So we took advantage of the Swedish national health system where everybody and every visit to the doctor is recorded in a national database, and we were able to estimate how much of autism is caused by these common things that we all have and these rare things that only some of us have, and then non-genetic things or things we don't really understand. And what's striking is that half of autism risk comes from genetic variation that we all have some of. Things that are common enough that everybody listening to this webinar will have a couple of them. And it's when you have too many of these variants, each one increasing risk by a tiny amount. When you have too many, that's when you become at high risk for autism. So there may be 1,000 or 10,000 of these things that we all have, but some people may have 10 of them, some people may have 100, some people may have 1,000, and then they're getting to the point of risk. And I'll give you an example of that in the next slide. And then these things over here, this 3, and 4%, this 10%, these are rare genetic variation of more significant effects. So the top left of that curve from before. And this 41% I'm calling unaccounted for. It used to be called environment back in the day when environment meant things that were not genetic really. Nowadays people think of the word environment to mean something out in the environment, like in the water, and that's not exactly what this means. These are things that are complicated. It could be genetic things like gene by environment interaction is over here. It could be mitochondrial things, mutations that are somatic like a cancer-like mutation. It could be many other things. So this area of autism risk is really underexplored and really we don't know much. What we know a lot about is this area over here, and that's why I'll be focusing most of my talk on the new findings in that area. Okay, so that's the common variants that are about 49% of total risk right here. So they're common, we all have a few, and they are of tiny effect. This is an example of schizophrenia, another very severe psychiatric disorder, and in schizophrenia, where they have about 25,000 individuals that have given DNA for this study and with schizophrenia and about an equal number of controls, they have found 108 of these common variants. These are chromosomes 1 through the X chromosome. Each green thing is a very significant finding of a part of the chromosome that increases risk for schizophrenia. So this is 108 out of probably a significantly bigger number, but you can see that some people may have just this one, some people may have this one and this one, but the more of these common variants you have, the greater your risk for schizophrenia is. And this is a way of plotting 3 populations based on whether they have the smallest number of risk variants or the largest number. So everybody was plotted out and then divided into groups into deciles. So this is the top 10%, or the low 10%, and this is the top 10%. And what you can see is that the people who have the greatest number of these common variants out of this 108, okay, have increased risk for schizophrenia of 7 to 15 to 20-fold compared to people with the lowest number of these variants. So this process of things adding together to cause risk is called additive genetics. And it turns out to be very important in virtually every psychiatric disorder. It's not just one thing, it's things adding together that cause the risk. Okay. And in schizophrenia and other disorders where we have this much data, people are now beginning to stratify people and say this person is at risk for schizophrenia. And that turns out to be very powerful because then you can say, all right, this person's 13 years old, at risk. First of all, we can monitor that person over time, and we may be able to start to think about interventions to prevent the onset of schizophrenia. All right. In autism, this is an autism study from the Psychiatric Genetics Consortium, there are not 25,000 samples. There's barely 7,000 yet. And as a result, we have only one genome-wide, this is a little bit out of date, this slide. We have only one finding for this common variation in autism. Over the next year, we predict that we'll have about 10. But it'll take quite a few years before we get to a sample size of 25 to 35,000 where we can find as many as you can find in schizophrenia today. So even though these common variants down here account for about 50% of the risk, we know very little about them. So now I'm going to switch to these over here, which are rare. These are genetic variants, so it's a gene that might have multiple flavors, if you will, and some of those flavors increase risk for whatever the disorder by many-fold. All right? And why is that so important to discover, and why is it really changing the nature of novel medicines for psychiatric disorders? If you have a psychiatric disorder and you understand, you discover what causes it, this says gene, and that's what we'll be focusing on, but it could be a non-genetic cause too, then you can do something quite amazing. You can take that gene mutation that you found in a human and you can introduce it into an animal, for example, a mouse or a rat, and I'll give you some examples of that as we go forward. So now that mouse or rat has the same human-type mutation that you'll find in an individual with that disorder. And now you can do something which is very hard to do in humans. You can study the brain in great detail. So you can put in, for example, electrodes in the brain. You can ask how different parts of the brain are communicating. You can ask how robust the brain is to various kinds of noise. And you can begin to understand more and more by studying that mouse brain what's gone wrong. What is the pathobiology or pathophysiology? This picture, this is a mouse brain, by the way. That's a human brain. That's DNA. This is the point of contact between two brain cells or nerve cells called the synapse. And you can actually measure this exact activity between two nerve cells in a mouse fairly easily. And so you can begin to understand what's gone wrong with communication in the brain. And that may well, as you work up this animal, lead to an hypothesis about a potential part of, this is an example of a protein in a cell, some protein that might be a new target for a drug development. And if you come up with that target, then there are lots of very good universities and biotech and pharmaceutical companies that will develop a drug that hits that particular target in the brain. And now what you can do with that drug that you now have a good reason to believe may be effective is you try it back in your mouse. The mouse has the human mutation. It has hopefully some of the human characteristics, behaviors. You studied it and you understand something about the pathobiology. You come up with a new target from that study, a new drug, and then you try it in the mouse, and if the mouse improves, then you're in a strong position to argue or to go through all the trouble and cost to try that same exact compound in individuals suffering from that disorder. So finding a gene, and particularly a gene of major effect, a gene where there is a profound mutation in humans and you can make the same mutation in a mouse, you have a very easy path to trying to come up with novel therapeutics. And I'll give you some examples. All right. So these are two studies that came out at the end of last year. One of them is from the Autism Sequencing Consortium, which I'm one of the leads of, so I'll talk about that in greater detail. And the other is from the Simon Simplex Consortium, which is a very excellent group of people who are working with the Simons Foundation to characterize about 2,500 families with autism that were collected. And that's Evan Eichler, Matt Stade, and Mike Wigler were the leads of the SSC sample study. And these two studies are really revolutionary. First of all, they're the largest psychiatric genetic studies for rare disorders in the planet. This had 15,000 samples. This had about 10,000. There was some overlap, a small overlap. So between these two, there's about 20-something thousand samples of families, either individuals with autism or their family members or controls. And this was used to begin to find these rare variants of major effect. So this is the ASC, and as I said, I'm one of the leads of it, so I can speak to it with at least some knowledge. And the other PIs are Mark Daly, Bernie Devlin, Catherine Reuter, and Matt Stade. And this is a summary of our study. And the SSC study is very similar. The results are very similar, and I'll actually show you what happens when you put them all together. So we took 15,000 individuals, of which this is a mom, dad, and a child, and red is an individual with autism. So these are families. And then we also took cases, and we took very, very carefully matched controls. So that's how we got to the 15,000 subjects. We did something called exome sequencing. This is a procedure where you take all those 22,000 genes in the human body, and you sequence them, and you get the exact base composition, and any change from the reference, if you will, is detected here. And then you can begin to ask, how are these cases, what genes are different between these cases and the controls, or this child and its unaffected parents? And so you begin to find autism risk genes, and then you begin to understand the biology. And what we saw in summary, this is a nerve cell in blue, here's another nerve cell in yellow, and these are those points of contact that I talked about called synapses. And we found, not surprisingly, that a big source of risk for autism were mutations in genes that are involved in nerve cell to nerve cell communication. That was kind of suspected or known. What was quite novel was we discovered that a large part of what happens in the nucleus during development, which is when DNA opens and closes, turning genes on and off, and then regulating how they're actually expressed as proteins, that turns out to be a major source of autism risk. So this is basically processes that happen during the development of the brain. Early development of the brain, in fact. Okay, so this is the blow-up. I'll show you this now in a blow-up form. Here is one nerve cell, here's the other. Okay, and there's a little gap between them where chemicals go. And all these genes that are found in this nerve terminal, or this area of contact between two nerve cells, we found mutations in them in autism. This is the other part of the equation. This is what happens in the nucleus, or the chromatin remodeling. And DNA, which is this little gray line here, is actually wrapped around protein. The proteins are called histones, H2, H4, H3, and that's a way of compacting the DNA, but also turning it off and on. So when DNA is very compact, it's turned off. When it begins to unwind, that's when that gene can express its protein. And this is one of those proteins, histone 3, and this is part of that protein. And everything in color, or even in gray that you see here, are enzymes that modify that protein at these various Ks, which is the amino acid lysine. And what you can see is that there are many, many genes that we found with mutations actually are involved in the modification of histone. And the modification of histone is what, in turn, turns DNA off and on. So during development, it may be that this part of the DNA right here has to be kept silent because it's not important during development. Or it may be that this part of the DNA has to be kept open because that's really important to making proper cell-cell connections. And so things that keep this open and this closed are going to be very important to be properly present and functioning. And so mutations in all the things that are responsible for that open and closed configuration of DNA turn out to be critically involved in autism. And the scope of this was really underappreciated until these two studies came out. We knew that there were a couple of genes. We didn't realize that there were dozens and dozens of genes that are involved in this process of opening and closing DNA that were involved in autism. In fact, these family of proteins, these lysine methyltransferases and lysine demethylases, there aren't too many in the human body, and yet many, many, many of them have mutations in autism. Okay. So those sample sizes of 15,000 or 10,000, they sound really big, but autism is very complicated, and these are rare mutations, and so it turns out that even that number isn't big enough to find all the genes. But it is big enough to begin to estimate the number of genes like this that we might find. And this is from Stefan Sanders from UCSF, and basically showing that as the samples got bigger and bigger and bigger and bigger, the number of genes we found followed a curve that was exactly like you'd expect if there were about 1,000 genes that contributed to autism in this way. And some people say it's 800, some people say it's 1,200, some people say that's not important. It's certainly not 5, and it's certainly not 50,000. It's around 1,000 plus or minus a bit. And once we know that, we can actually predict what sample size we need, how many families have to participate before we find most of the autism genes. And in autism, we're still a ways away, but in other developmental disorders like intellectual disability or learning disability, we're actually much closer because there are fewer genes, and I can predict that in the next few years we will probably have found most of the genes, at least the dominant ones on the autosome to be for the specialists, that underlie intellectual disability. I'll give you an example of that later. So we think there's about 1,000 genes that contribute to autism. And when we look at the genes and we lump them together, this is from Catherine Reuter from Carnegie Mellon, the effect sizes for the mutations we're looking at are on average 20-fold increase in risk. And some of them are up to about 80-fold increase in risk, and some of them are between 5 and 10. But on average, the most significant mutations we're seeing that are point mutations have about a 20-fold increase in risk. That means that if you assume that the population prevalence of autism is 1%, having this one mutation will increase your risk to 20%. That's profound, right? And as I said, there are a few genes we found, and only a few, that have risks of closer to 60 to 80-fold. By that point, 60 to 80-fold is very, 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 very high risk for autism. Okay. So I've talked about these common variants, which we don't have many of, but we know are important. And I talked about the rare variants, which we have some of, but we know we need to find more. I've also showed you that in schizophrenia, the example I showed, it's an additive genetic system where things have to add together to produce the final risk or manifestation of disease. And in fact, in the same study I talked about earlier in Sweden, we showed very clearly that in most cases of autism, it is additive, and more importantly, it is the interaction of the common and the rare genetic variation. What that means is that it's not just this or this, it's this plus this. And one way to think about it is that the family, as do we all, has lots of common variants that increase risk for autism, and then the individual in the family gets a mutation that pushes that individual over a threshold to manifest with autism. One thing that's become very interesting and heavily studied in psychiatric genetics is that it turns out that some of the findings in autism are also being made in schizophrenia. Some of the findings in schizophrenia are also being made in bipolar. And so even though we consider these disorders quite distinct, the genetic risk factors seem to overlap to some degree. And one hypothesis is that, if there's a family that has lots of common variants associated with autism, then when they get this rare mutation, they get autism. But if that same family has lots of common variants associated with schizophrenia, when they have the same rare mutation, they can manifest with schizophrenia. That's an hypothesis that really is just that, but we're far away from proving it. But it does begin to tell us something about how genetic risk for psychiatric disorders actually overlap quite substantially. All right, so this is in the paper, and it's a little bit dense here, but basically we said that if there was, for example, an individual in the family that had a new mutation, a spontaneous mutation of one of two kinds, and they didn't have that mutation, 80% of them would not have autism if they had a major mutation, and 60% of them would not have autism if they only had a smaller or less profound mutation. In other words, they have this risk that comes from the common SNPs, then they had an added mutation, and it's that mutation that if you took it away, there would be no autism, even though the risk is still there. So that's kind of the interaction of the common and rare and the inherited and spontaneous mutations. All right. And this is what I kind of intimated earlier. As we find genes for autism, they're also the same genes are also genes for schizophrenia. So each one of these things is the name of a gene. Very exciting names like MYH10 or POGZ or AXL. And all of these genes that are here were found in our autism study, but everything in this circle was also found in schizophrenia studies. Everything in this circle was also found in intellectual disability studies. This is genes also found in congenital heart disease, epilepsy, and metabolic disorders. So we're confronted with this, what I mentioned already earlier, that nobody's going to confuse congenital heart disease with autism or even schizophrenia with autism, unless it's atypical. And yet the gene findings are overlapping. And that goes back to the question of how does that happen? And as I kind of intimated, it might be the interaction of common and rare variation. Okay. And so this turns out to be not, I mean, as you can already see here, it's true for many of the disorders that are being studied now. Schizophrenia, intellectual disability, epilepsy, autism, okay? There is something called many-to-many, which is what every database person hates because it's actually impossible to code many-to-many relationships in databases. But here's autism, autism spectrum disorder, intellectual disability, schizophrenia, epilepsy, congenital heart disease, MCA, multiple congenital anomalies. And in autism, all these blue lines are links between the disorder and genes. These are those gene names again. And these genes are ranked by how we found them in our first study. So CHD8 is an autism gene, but it's also a gene implicated in congenital anomalies. SNC2A is an autism gene, but it's also an intellectual disability gene, a schizophrenia gene, and an epilepsy gene. And ditto as we go down. So every disorder on this side has multiple genetic causes, and virtually every gene on this side is associated with multiple disorders. So this many-to-many relationship is troubling, exciting, confounding, you name it. It does have some some good parts to it. One of them is that, and I'll show you later, once I know this, I can put together all the data from this, and maybe even this, and find more genes for all of them. Also, if in the course of the next few years somebody comes up with a drug for this mutation, it might help people with autism or intellectual disability or schizophrenia. Right? So it is a profoundly complicating thing, but it does have some benefits, and as I said, I'll give you an example. So these are the two studies I talked about earlier, the Autism Sequencing Consortium and the Simon Simplex Collection. And once all the data was organized and available, the two groups took the logical step of putting all the data together. So this is almost 20,000 samples. And Stefan Sanders, who I mentioned earlier, was the lead author, and from Matt State's lab. And many of the senior people in the field actively participated. The ASC was a formal collaborator on this study. And the main findings were the synapse genes and the chromatin, the DNA unwinding genes. So nothing new emerged, so to speak. More genes emerged, but the general picture remained more or less the same. But we have a few more genes now. In fact, we have 71 genes from this combined, or 65 genes and a few other large gene loci. So we're getting more and more genes as we go forward. All right. And this is so this is putting together the ASC and SSC. This is putting together the ASC data and data from the Deciphering Developmental Disabilities Consortium, which is a consortium that's founded in the UK. Jeff Barrett and Matt Hurls are two of the leads of it. And they have been sequencing 12,000 individuals with intellectual disability. And this is just the first, just over 1,000. So we took our autism data and the intellectual disability data and we put them together. And lo and behold, we found, as would have been predicted from this thing, putting this with this, we found more genes. We found 16 genes that were not found in either data set by itself. We also began to see that genes that were found both in the autism and intellectual disability sample had a profound effect on IQ, as did the intellectual disability genes. But genes only found in autism and not in the intellectual disability sample had a much more modest effect on IQ. So we are beginning to tease this complexity apart and say there are some genes that are not ID genes. There are some autism genes that are not ID genes. And of course, there are a lot that overlap. So we're beginning to get some evidence of specificity for the first time. We're saying this is an autism gene, a non-intellectual disability gene. This is a gene that impacts both. All right. So that's kind of the framework of what's going on in gene discovery in autism. And then I'm going to go to a specific gene and a specific example and show you what we can do. What I showed schematically, I'm going to show now very specifically in a disorder called PMS. And the families that are that have this kind of disorder have created a tagline for PMS. It's not what you think. So I can't take responsibility for that. It's a great tagline. PMS stands for Phelan-McDermid syndrome. These are the two people who first described the syndrome, and so it's named after them. It's a genetic disorder that has a very high risk of autism, and it's caused by mutations in a gene called SHANK3. That's the name of the gene. All right. And how common are SHANK3 mutations? It turns out that SHANK3 mutations, which we really didn't even know about till recently, account for as much autism as Rett syndrome and maybe even as much as Fragile X syndrome. If you look at individuals with autism that are more severely affected, about 2% of them have a mutation in this single gene, SHANK3. And this is one of those genes that I said probably increase risk by 60 to 80-fold. Okay. We've done a very large prospective study of individuals with PMS, and I can tell you that they have autism. 80% of them have autism. I can tell you also that they have typically severe to profound intellectual disability, so they're very severely affected. They typically don't have language, and the estimate, a conservative estimate, is that it's 1 in 10,000 individuals. It's probably closer to 1 in 5,000, and that's in the order of Fragile X syndrome and Rett syndrome. And I'm sure you've heard of at least one or the other of both of these, or you may have. And I bet that many of you haven't heard of this disorder, PMS. And so that's what's amazing. A disorder that's as common as certainly as common as Rett syndrome, still is a fairly recent discovery. And this study that I showed you was the first ever prospective study of individuals with this disorder on a larger scale because it just was unknown. Okay, so back to this question of benefits. I mentioned a couple of them about benefit to family, and let's take a look at these two, recurrence and family and advocacy group and clinical course. Okay, so this is a reality of Phelan-McDermid syndrome that about 25% of cases come from a parent with a balanced translocation. What that means is here's two chromosomes. We have 23 chromosomes, and we're calling this chromosome A and chromosome B, and they're different sizes and different colors to keep it straight. And it can happen that your chromosomes during development, pre-development, can actually break and then reform and be perfectly fine. And even if chromosome B is now on A and the end of chromosome A is now on B. No genetic material was lost, it just ended up in a different place. But if that person with a balanced translocation has a child, and for example, here's that person with their chromosome A plus the B and chromosome B plus the A, and this is the chromosome B that this child got, now it doesn't get this anymore because it got this chromosome from mom and this chromosome from dad, and so it doesn't have this extra material which it should have had because this piece of chromosome that it got, this hybrid chromosome AB, does not have this part of chromosome B. So now the child is missing a critical DNA. Mom or dad were not, but the child is. And since this will happen 50% of the time, this individual makes a sperm or egg, 50% of the kids from this individual could have PMS if the deletion is on chromosome 22. So this is what I mean by recurrence. That family can now be told something very specific. You have a child with Phelan-McDermid syndrome, and we need to tell you that you have a 50% chance of having another child with that disorder. And so that family can make informed decisions about counseling and genetic testing. Okay, they can also go to the web and Google Phelan-McDermid syndrome, and they can find a foundation called the Phelan-McDermid Syndrome Foundation that has a meeting every 2 years where all the families get together. It's done with the kids there. It's down in Orlando and done around Disneyland, and the kids love it, families love it, and they get to all meet each other. They get to they get to learn from each other. They get to raise money. They get to figure out how to be more vocal advocates to get more money into research. They certainly contact organizations like the BBRF and say, we'd like to see more research going on in autism or intellectual disability or Phelan-McDermid syndrome, and they kind of help set the stage for the ultimate improvement of treatment for their family members. So they become active participants in this important process. Here is the PMSF Facebook page. So, and by the way, when they talk to each other, they learn about clinical course. Right? So these are ways in which the families benefit immediately when there's a genetic diagnosis in this case. All right, so let me move to the other more nebulous, if you will, overall benefits to society and how that feeds back to what might be called personalized medicine. That means giving a treatment to an individual based on the cause of their disorder. So you personally find out what's going wrong and you treat that. All right. So this is the SHANK3 gene, and these are the various mutations that have been found. And I'll just to say that there are a lot of mutations in this part, in the red part of the gene. So we took that same red part of the gene in a mouse and we broke it. We made it have like a human mutation. And these mice, okay, spend less time. This is the typical mouse, this is the mouse missing the gene, one copy of the gene. They spend less time in social interactions, whether it's by sniffing each other or by making ultrasonic vocalizations, little squeaks that you and I can't hear. So their behavior has changed. Certain connections in the brain. This is measuring the connection between two nerves and brain cells. This is how much energy was put in, and this is how much energy came out from the downstream nerve cell. And what you can see is in a typical mouse, as this goes up, as you put more electrical current in on the upstream nerve cell or brain cell, you get more of a response from the downstream brain cell. But this other, in the heterozygotes, I'm missing one copy of the gene, so we've knocked out the red part of the gene in a mouse. It plateaus very quickly, and you put more and more electricity or energy into the system, and there's no more increase in the downstream cell. That's a bad thing. That means that the critical connection between brain cells that helps us do everything, including learn, remember, move, is broken down in these animals missing just one copy of the gene. So that's bad, but we know exactly what proteins underlie this, and they're called AMPA receptors. And we already know, and I won't show you this, we already know that if we give these same mice something that enhances those receptors, then they look just like they should. So doing a little bit of biology, and you already have a lead for a possible new drug. The drug we started out with was a drug called IGF-1, and we started for a variety of reasons. One is it already is being shown to be working in Rett syndrome, which is another autism-associated disorder, and wouldn't it be nice if one drug works in two different autisms with different causes? The other reason that we tried IGF-1 in our animals is that it is goes into the brain, we know that. It is very safe. It's used extensively in kids with a certain kind of short stature syndrome. They can use it for 6-8 years. We have enormous safety data. in kids, which is where we really want to do these kind of trials if we can. And as I said, in Rett syndrome, it has beneficial effects in a first small trial. So we tried it in our mice. So remember, the mice are missing part of the SHANK3 gene. And this is another way of looking at the connection between nerve cells. This is the before, and this is the upstream and downstream. And this is a form of cellular memory. So the relationship between the two cells, we call it 100% at baseline. We give a stimulus, and if you look at the blue, in a healthy mouse, the connection between those two nerve cells is now increased for days to 50% or more of what it was before. In other words, you have reinforced that connection. So if it's, for example, a connection involved in remembering something or playing a certain piano piece, okay, this is a cellular trace of the memory that's being formed, and this, as I said, will last for 24 hours or more. Missing just one copy of the SHANK3 gene, and the memory is made, but it decays within 90 minutes. However, if we take that same mouse or its brother or sister, also missing one copy of the gene, the het, and we give it this IGF-1 at a clinical dose for 2 weeks, you see that now it looks exactly like the wild type or typical mouse. So we've reversed a kind of a memory deficit in the mice. The mice also show motor abnormalities. If you put them on a rotating rod, they fall off more quickly. So this is the typical mouse. Every day it gets better. It learns how to roll on this log. Missing one copy of the gene, it falls off more quickly at 100 seconds rather than 120. And if you've given the animal this IGF-1 drug, it does at least as well as or even better than a typically developing mouse. So we have a drug that we know is safe for use in kids, and it reverses cellular changes in the brain and behavioral changes. And so we did a very small pilot controlled trial of IGF-1 in this disorder in kids now. And why is the figure not here? Well, you have to trust me. There was evidence from 2 measures that the kids got better in a 12-week treatment. They got better in terms of their social withdrawal and in terms of some measures of repetitive behavior. It's a tiny study. It was just 10 kids. It was kind of a preliminary safety efficacy study. It has, not surprisingly, generated a lot of interest. This is the first clinical trial of any kind, certainly the first controlled trial in Phelan-McDermid syndrome. And so when you have a finding like that, you know, it generates a lot more excitement. So I'll kind of go out of order here. NIH gave us resources to make a second study of it. The company that makes the drug is talking to us about reducing the cost. The reason we only did 10 at the beginning is because it costs us $14,000 for drug per patient for just 12 weeks. This is going slower than I'd like, but the company is interested now because this is a market for them, and of course they're also interested in helping people. We have reached out. It's a rare disease, and we want to make sure that everybody knows what we know. So the NIH has actually put together and funded a rare disease clinical research network that it looks at 3 genetic causes of autism, and there are now 6 sites within the United States that work together on this network. We all try and do the same things and learn from each other, and Alex Kalevzon here at the Seaver Autism Center is the head of this kind of consortium, the Phelan-McDermid syndrome part of it. We spend a lot of time telling other people, like EU-AIMS is a major European consortium that we share our data with. We've gone to Spain many times to talk to the people there and to work together, many times to France, Germany a few times. So we try and disseminate the information and learn from each other. And then, you know, I started this by saying IGF-1 works in Rett syndrome and may work in Phelan-McDermid syndrome. So, of course, the next obvious question is, will this work more broadly in autism, even if you don't know the cause? And so the Autism Science Foundation wants us to answer that question and has given some money already to start looking at IGF-1 in autism where we don't know the cause. Idiopathic autism just means autism where we don't know the cause. So we're starting from these genetically defined causes of autism like Phelan-McDermid syndrome or Rett syndrome, but the ultimate goal would be to find drugs that work as broadly as possible. Okay, I'm going to finish up pretty quickly and say in the same way we can mutate genes in mice, we can mutate them in rats. Rats have an advantage that their behavioral repertoire is much broader, and the neurobiology, the study of the brain is much more well characterized. This was funded by parents who wanted to see this work go forward. And one thing we can measure easily in rats and not so easily in mice is attention. This is basically a measure of something like ADHD. And we know that in autism, and particularly in Phelan-McDermid syndrome, there are major attentional deficits. And so this doesn't look like much, but the animals missing one or both copies of SHANK3 have an attentional deficit. They do less well in choosing the right thing that lit up compared to the animals. They omit more, they take longer to come to the right decision. So this is actually showing for the first time attention, like ADHD-like symptoms in a rat model of autism. The other thing we saw in this rat model was a deficit in social recognition memory. So let me explain that quickly. If you put two rats together, a gray one and a black one, for an hour, and then you wait some period of time, either 1 hour or 24 hours, they will, and then you put the gray and the black and a new rat, a white mouse rat together, the gray rat will spend more time with the white rat. So it met the black rat yesterday or an hour ago, and now it meets a new rat, and in many kind of very hierarchical animals, they spend a lot more time with a new animal than with the old one, because they already have a relationship established there. So now they're spending more time with the new rat. And so, for example, after 1 hour, this gray rat spends much more time with the unfamiliar rat compared to the familiar rat, the white rat versus the black rat, whether it has both copies of the gene, a mutation in one or two mutations. But after 24 hours, the typical rat with no mutation still remembers that it met this guy yesterday and will still spend more time with the new rat. But missing one or two copies of the gene, they're no longer able to remember that they had met this rat yesterday. What's so interesting about this very specific paradigm is we know exactly, first of all, it's only for social, it's only for rats, it's not for objects. But we know that this whole paradigm is regulated by a peptide called oxytocin. And so, of course, we did the obvious thing, which is we took animals with a mutation, either in one mutation or two mutations, and we gave them oxytocin. Without oxytocin, with just salt water, they didn't remember the next day. No difference between how much time they spent with the new versus the old rat, or the familiar versus the unfamiliar rat. If they had been given oxytocin at the beginning, they remembered perfectly who they met, who's new, and who they met yesterday, whether they had one mutation or two mutations. And so it won't surprise you to know that, again, oxytocin, we chose it because it's very safe. And so we now have FDA approval to try this drug, oxytocin, in Phelan-McDermid syndrome. All right. And finally, and I won't belabor this, we have a third lead from these animals for another kind of drug. So all that's to tell you that, when you find a cause of a disease, you can begin to understand, to model it, and begin to understand what's changing in the brain, and that can frequently lead to novel drug targets and novel drugs. This is something that's never happened before in history of drug development. The idea that we're using neurobiologically driven approaches to come up with new drugs is a completely novel approach, and it's really taking the greatest steps forward in autism and developmental delay syndromes generally, and there are many examples. I gave you one, and I gave you some examples in Phelan-McDermid syndrome, but there are examples in Fragile X syndrome and tuberous sclerosis, even in Down syndrome, and so I think that the people like me who are in this field feel that we are actually leading this new paradigm in drug discovery, and since there are so many irons in the fire because of all those genes, there are so many possible targets, I actually feel confident that we're going to start getting some successes in the next few years of completely new drug approaches to treating autism, intellectual disability, and associated disorders. So to kind of summarize, there are many autism genes, that's the bad news. They're associated with very broad spectrum. Some of them are genes of minor effect or major effect. Most cases are additive. Multiple things work together to cause the disorder. The genes that we know so far map to the contacts between nerve cells, between the chromatin, which is that thing that turns genes on and off, and transcriptional pathways that again regulate genes and turn them on and off. This many-to-many relationship that each disorder has many genes, each gene is associated with many disorders, seems to be true for many things. But we can begin to tease these things apart. And this is, I think, the most exciting thing. There are many examples, I just gave you one, of genes that then led to novel clinical trials in autism and developmental delay. And I think over the next few years we're going to start to see whether this approach, this hypothesis that I put out in the second slide, is real. That if we know etiology and we know causes and we can model those causes, we'll come up with new drugs. If you want more information about the CBER Autism Center, that's our website, cberautism.org. We have a summary of the work we're doing. Every year we put it out. That link will have it. If families are interested in participating or people want to come volunteer or work in the CBER Center, that's where to contact. Autism Sequencing Consortium is a brief write-up on the Emory website. And if people want to participate in autism research through an ASC site, if you contact Jessica Brownfeld, we will point you to the right site for participating in research. And just to show you, this is the ASC. Remember I said we had a publication with 15,000 samples. We have now 22,000 in the current data freeze. We expect to have 35,000 in the next 12 months. And as you can see, we're getting samples from all over the world, from major groups within the U.S. like the UC Davis Group, University of Miami, but from Sweden, Italy, Hong Kong, Brazil. There is so much interest from the funding agencies around these kinds of studies that they are giving us the opportunity to take samples from anywhere in the world and to sequence at least up to 5,000 a year through these central sequencing sites. So if families interested in participating, this is the place. This is one way to do that. So I want to thank you, and I know I ran a little late, but thank you for your attention, and I'll stop there.
Joe, thank you for an extraordinary presentation, and I think running late was well worth it because as you kept going, you answered a number of the questions that came up, and probably at the end the most frequent, which is, where can people go if they want to get involved? And we will have this information up on our website as well so that people can go back to it and follow up. I want to emphasize what you emphasized, which is the excitement of the approach, that instead of finding out that some medicine helps a particular disorder almost by luck, as was the case in the past, you're actually doing very basic research and using that basic research to develop actual treatments that can be used in real people. And that's a different approach from what's happened in the past, and very, very exciting. And a question that I have is, in addition to these treatments that are potential treatments for people who may have the condition, are you finding treatments that could potentially prevent the illness from happening in the first place?
So, and that all comes back to the question of, early diagnosis, right? So when we do have a genetically determined cause of autism, we can study that individual from birth, right? And we can watch as things progress, and we can also think about intervening to prevent, right? So in other words, in order to prevent, you need to diagnose before onset. And so in the behavioral world, people are getting better and better at beginning to pick up signs of autism at 12 months or even earlier, and those are certainly going to lead to attempts at intervening early on before the manifestation of symptoms. And it's also true in the genetically defined cases where, if you know, for example, some of these genes are passed down within the family, and so we can actually predict who's at risk, and there are people beginning to think about doing interventions before the manifestation of the disorder. We're not there yet, but it's certainly something that we're now able to do because of earlier and earlier diagnosis before this full manifestation of symptoms. The flip question, of course, is what if somebody already is older, already has a diagnosis, can we turn back the clock if so many of the genes are involved in early development? And that is, remarkably enough, an area of active study throughout the world where people are turning these genes on and off in mice, which you can do it in mice, and saying, oh, look, if we turn it back on in the adult mouse, the mouse recovers much of its lost function. And there are even people, and Takeo Henshin, one of his proteges, right here at Mount Sinai, who are actually figuring out how to make the brain go back to its kind of more plastic state of early development and become amenable to being retrained. So if there's something even later in life, we do think over time, not today, but as in the years to come, we'll be able to actually reprogram the brain to kind of redevelop wherever there are problems. I didn't say that very articulately, but
but you said it in a way that I think people can understand, and that certainly offers a tremendous hope to people. And I think a key message is for people who may have a concern about their child, immediately go and have an evaluation, because early diagnosis is extremely important.
Yeah,
go ahead. Yeah.
And the interventions that we know work, the behavioral ones, all the evidence suggests that sooner is better.
Right.
Later is not too late, but sooner is better.
Yep. And that there is always hope and help available, and people should seek that.
Yes.
Our time is up, so I want to, Joe, thank you not only for your presentation today, but for the extraordinary work that you have done and you continue to do in this important area. I also want to thank everybody who joined us and want to remind people that the research that we fund is made possible through private donations. So if you'd like to make a gift, please visit bbrfoundation.org or call 1-800-829-8289. This webinar has been recorded, and if you've missed any portion of the presentation or would like to share it with a family member or a friend, visit the webinar page on our website. We will also have on the website a listing of our upcoming webinars in 2016. And let me take this opportunity to wish everybody a happy holiday and a happy and a healthy new year. Once again, thank you all very much. Take care.
Thank you, Jeff.
Thank you.
And thank you to the participants, too. Happy New Year, everybody. Bye-bye.
Take care. Bye.